and welcome to the session number 7 of the AtC-AtG conference. I'm Ksenia from Immanuel Kant Baltic Federal University, and I will be chairing this session together with Ilona Lucerf from National Institute of Applied Science in Toulouse, and Jack Harrison from the University of Oxford. And we will start from 30-minute invited talk by Mariia Efremova, and then we will have 6 oral presentations. And I would like to remind that if you have any questions, you can write them on the chat, and you can do this during the talk, or raise your hand in the end of the talk, and we will unmute you. So also you can explore the Gather Town, where you can visit the poster session, chat with our invited speaker at the bar, and connect with other people. So it's my pleasure to welcome Mariia Efremova. And Mariia obtained her PhD at Lomonosov Moscow State University in Russia. Then she was awarded a long-term fellowship from the Federation of the European Biochemical Societies, and then a Humboldt Research Fellowship for postdoctoral researchers to conduct research at the Technical University of Munich. And this year, Mariia received Eurotech Postdoc 2 Fellowship and joined Eindhoven University of Technology. So with currently working. So Mariia, can you please share your screen?
Yes, thank you very much for kind introduction, Ksenia. So let me start the presentation. Can you see the slides and hear me clearly?
Yeah, everything is okay. And you will have 25 minutes and 5 minutes for questions.
Thank you very much. Welcome, everyone. Thank you very much for attending this presentation. This is my first global virtual conference, and I would like to sincerely thank the organizers for inviting me. Today, I will introduce you to the field of biomagnetism, or genetically controlled magnetism. It's pretty different from, for example, synthesizing chemically magnetic nanoparticles for further biomedical applications. Instead, we actually don't synthesize anything in the lab. We study what Mother Nature has created for us in terms of biomagnetism, and maybe only guide it to the necessary direction. This branch, opposed to other magnetism fields, is relatively new. So I hope you will find it interesting and inspiring. Let's go to science. But before, I would like to just briefly say that I here represent the Eindhoven University of Technology in the Netherlands, where I'm currently doing a postdoctoral project at the Department of Applied Physics in the group of Professor Dr. Reinhard Lavrijsen. And at the very end of my talk, I will give you one slide teaser on the project which is currently running. But today, I present the results which were obtained in Germany during my first postdoc at the group of Professor Dr. Jill Westmeyer and the Technical University of Munich, in collaboration with the University of Duisburg-Essen and Herkules-Zentrum Berlin in Germany. Some of the results were obtained at the site of our collaboration partners at Lawrence Berkeley National Laboratory and United States, and National University of Science and Technology Mises in Moscow, Russia. What about biogenic magnetic nanoparticles? The most famous one you have probably heard about occur in magnetotactic bacteria. They represent magnetite Fe3O4 or greigite Fe3S4 and have ferri or ferromagnetic properties. These particles have a well-defined crystalline structure, and as you can see on this nice image on the left, can even have facets around them. They are formed in a special cell compartments, which are called magnetosomes. In terms of magnetical properties, these particles are pretty well characterized, and also we know very well how exactly these particles are formed in bacteria. But what about mammalian cells, so the cells in our body? The iron storage in mammalian cells is mostly represented by ferritins, which store phosphorus-rich ferrohydride with the formula presented on the slide. Ferrihydride has antiferromagnetic properties, and the particles inside ferritins can be either amorphous or poorly crystalline. Since the ferritin is a nanocage, iron is primarily stored inside its cavity. Characterization of ferritin is pretty challenging though, primarily because of its low stability, either chemically, so in the change of temperature or pH, or also during the measurement itself. For example, the irradiation of X-ray beam in some important techniques. Still, the characterization of ferritin is very, very attractive to scientists, because under some very specific conditions, it can be transferred to magnetite. And the famous examples would be the teeth in chitons. Chitons are mollusks, and in young species, their teeth mostly contain ferrihydride, while adult species, under the letter D, they have magnetite. So somewhere in between, the transformation happens, and still no one actually knows how exactly it's going. What else about ferritin? It has been proposed for cell manipulation, namely by triggering so-called ion channels, which are the gate of ion influx inside and outside of the cell. Ferritin can either create local heating or force or a torque created by pulling the ferritins with external magnetic field gradients. However, after a couple of successful examples shown in the literature, there was another paper showed by physicist Markus Meister that simply given material inside of ferritins and its size, he calculated how big magnetic moment can be that ferritins generate. And he showed that these forces are simply several orders of magnitudes too little to generate enough magnetomechanical response from the cell. Therefore, no successful cell manipulation example with ferritin, or maybe convincing to be precise, has been shown so far. But let's have a closer look at ferritin. This is a rather small nanocage with an inner and outer diameter of 8 and 12 nanometers respectively. However, ferritin is not the only nanocage that Mother Nature has created for us. In some bacteria, you can find encapsulins, which are basically ginormous ferritins with the inner and outer diameter up to 35 and 43 nanometers respectively. Yes, I said bacteria. However, if you take the DNA from bacteria and put it to mammalian cells, you can also make encapsulins there. And no one has ever studied the physical properties of encapsulins before, which brought me to the motivation of my research to create physically plausible biomagnetic interfaces for cell manipulation based on encapsulin proteins. This is the outline of my talk. I will briefly explain how these proteins were obtained in mammalian cells and iron-loaded. Then I will touch the phenomenon of magnetic cell sorting. We will go through structural and elemental composition study of nanoparticles, as well as their investigation of magnetic properties. Finally, I will show you an example how these encapsulins can be used for magnetic cell manipulation. As I said, we took the DNA from specific bacteria and added it to a model mammalian cells we chose for experiment. Afterwards, we added iron 2+ salt in the cell environment. And 24 hours later, the iron oxide accumulation inside the encapsulin cavity started. I would like to briefly point out that the cells also contain fluorescent protein scarlet-1, which gives quite intensive red signal. This will be important at the very end of my talk when I will say about magnetic cell manipulation. But just remember that these cells are also fluorescent, which doesn't affect other properties. 48 hours after we added the iron 2+ salt in the cell medium, we performed so-called magnet-activated cell sorting, which is a standard technique used for cell sorting, and this is obviously based on their magnetic properties. One use for it commercially available magnetic columns, which are fully packed with magnetic microspheres. When you pass the cell population through this column, the cells which have ferromagnetic properties and room temperature are stuck, just because the whole construction is put inside the permanent magnet. But the cells which don't have pronounced magnetic properties at room temperature can freely pass through. Therefore, you can separate the cells based on their magnetic response, again at room temperature. I would like to point out that normally people use chemically synthesized magnetic particles to label the cells. In our case, we did not add any external particles, but only relied on natural iron biomineralization inside the encapsulating nanocompartments. The fraction which was retained inside the column, I will always call the sorted cells, while the fraction which really passed through, I will always call the flow-through. So what's about the efficiency of sorting? So how many cells we actually could retain inside the column? It was not very high, actually it was pretty low. We could only sort from 1 to 2% of the whole cell population. Nevertheless, this was statistically significant and reproducible. And also, when we eliminated magnetic field parameter, when, for example, I only hold the column inside my hand instead of magnetic rack, we couldn't sort any cells at all. Therefore, there is something special about the cell population, which gives it a higher magnetic response. And of course, we're very curious what exactly is special. I started very simply, just looking at the cell pellets. So just the clump of the cell, I ended up after magnetic cell sorting. And as you can see, the sorted cell fraction has pronounced red color. So, and we remember that red color corresponds to M-scarred fluorescence. But that was just the first observation. More importantly, when I dissolved the cells and measured the iron concentration by inductive coupled plasma mass spectrometry, turned out that the sorted cell fraction is iron-rich compared to other cell fractions like flow-through, which cells which were not retained in the column, and the control cells which didn't express any encapsulins at all. So they are iron-rich. But is this iron actually in encapsulins? I would say yes, based on the results from transmission electron microscopy. So we took the cells and fixed them and ultra sliced them with two 70 nanometer thick cuts, which were further investigated by microscopy. Each electron-dense dot you can see on the slide refers to individual encapsulating nanoparticle. And we could find two types of encapsulating organization inside the cell, so-called dispersed, which are homogeneously distributed within the cell itself, or in some cases, they could form so-called aggregates. And I would like to point out that they were not just clumps of particles. They definitely had some kind of order inside. And in some cases, when there was some sort of intracellular support, these aggregates could even reach up to a couple of micrometer in size. And again, you can see a nice linear organization of particles, or also hexagonal symmetry between them. Of course, we propose that what if this is somehow related to their magnetic properties? But first, we decided to see what kind of structure these particles have. And turned out in high-resolution TEM and FFT transformation that these particles are amorphous, and only a very small areas, up to 10th, maybe hundreds of atoms, have some kind of crystalline order. And these nanocrystallites are shown by red arrows on the slide. From the viewpoint of elemental composition, we studied by EDX elemental analysis. This particle did not only contain iron and oxygen, but also substantial amount of phosphorus. At this step, we were really puzzled how these amorphous, low crystalline particles, also phosphorus-rich, can give us some ferromagnetic signal at room temperature. And of course, one other complication was that we could not further study the structure of particles by standard techniques like X-ray diffraction. Nevertheless, even the XRD was not available, we could still study local coordination of iron atoms. We first came up with the idea of X-ray absorption spectroscopy, which was done at the synchrotron radiation facility at Berkeley National Laboratory, where we studied the same cell sections we used previously for transmission electron microscopy, but this time they were studied by scanning transmission X-ray microscopy to find the region of these encapsulating aggregates when there are a lot of particles collected in the same place. And we recorded the spectra exactly from this area. The spectra from encapsulins was also compared to the reference spectra of control substances recorded at the same synchrotron station. As you can see, the spectra from our sample can be the best fitted with the iron 203 spectra compared to with two other standards. This is all good, but actually two phases of iron 203 exist in nature. One of them is alpha iron 203, which is antiferromagnetic hematite, and the second one is gamma Fe203, which is ferromagnetic mechimite. What phase exactly do we have? To answer this question, we turn to Raman microspectroscopy, which relies on the scattering of laser light interacting with the chemical bond between iron and oxygen. And we are interested, of course, in Raman scattering and how it resonates with the oscillation of this chemical bond. In the Raman spectrum, hematite and machimite give two pretty distinct sets of peaks. Therefore, there is a possibility to distinguish between them. For this experiment, we used even live cell, but again under microscopy control. This time it was light microscopy. We selected two areas. One of the area contained aggregate of encapsulin particles, and the second one was a free cytoplasm. One feature, of course, that strikes your eye that we have pretty high signal background, which could be easily explained by the fact that this is a living cell and contain many other chemical bonds which also interact with laser light. Nevertheless, these two spectra definitely differ at least at one position, which is approximately 700 reverse centimeters. We believe that this peak corresponds to the superposition of two peaks of machimite shown on this slide. And importantly, no peaks of hematite would be observed. For example, 607 reverse centimeters was definitely not there. Therefore, we concluded that this spectra we saw also together with X-ray absorption spectroscopy technique refers to gamma Fe2O3, so ferromagnetic machimite. This result was further verified by direct magnetometry measurement done first in vibrating sample magnetometry. We compared the magnetic signal from different cell fractions, and only magnetically pre-sorted cells gave us a pronounced hysteresis, while the other cell fraction like flow-through, the cells before sorin, or control cells with no encapsulins gave us only signal at the level of background. As you can see, the signal is pretty small though, and we use the second technique called ferromagnetic resonance to make sure that our data is actually correct. There we could find an angular dependent feature and could extract the absolute value of magnetization. This value we used to normalize the magnetic signal from vibrating sample magnetometry and obtain saturation magnetization. Surprisingly, we ended up with a value of 40 amperes square meter per kilogram, which matches very nicely the value for nano-sized machimite. Therefore, again, we concluded that we, first of all, indeed have machimite, and secondly, that it has ferromagnetic properties at room temperature. So this is more fundamental study and obviously very nice, but what about real application of these encapsulins? Can we do something useful with them in external magnetic field? I started with a pretty simple experiment where I took 3 different cell fractions. One of them was the sorted cells, the cells which were retained in the column before, the flow-through, which freely passed through, and there was also fluorescent control, so the cells which only expressed the fluorescent protein. So we remember that red color corresponds to the fluorescence of the protein M scarlet I told before. I took the suspensions of these 3 cell fractions and put it into separate wells of well plate. And I also put a small, in each of these wells, I put a small iron neodymium boron magnet, cubic one, and left the whole system overnight. When I came the next morning, I saw the pattern you can see on this slide, that the sorted cells nicely arrange themselves at the edges of the permanent magnet. While in case of the flow-through cells, we could still see some colocalization, but in case of fluorescent control, they were basically randomly organized, so no attraction happened. Therefore, it was a proof of principle that we can manipulate with the cells with a cubic permanent magnet. But this was a one-point measurement. Then I turned to a bit more sophisticated setup where magnetic needle was used. So I used the permalloy rod, which was connected to a permanent magnet, this disc-shaped one, through the permalloy cone, which was focusing the magnetic field. The end of the needle was immersed into the cell suspension, and as you can see, the whole system was built under the control of fluorescent microscopy. The needle can move up, so manipulator, this needle is located, can move up and down. Therefore, we can immerse and took out the needle, and all of this one can record on the video. Since we use the objective with water immersion, the objective was inside the cell suspension and could detect red fluorescence. So what did we end with? before I start the video, I just want to make a couple of clarifications. So you can see the outlines of the needle, more or less. It's here where my mouse is. And each bright dot here represents a separate cell. As you can see, even before I immerse this needle into the cell suspension, some of these cells already were attracted. So that's how the gradient was. And of course, we need to look at the tip of the needle where magnetic field gradient is the highest. So please look more in this area. So this video is 5 times accelerated. So in fact, cells didn't move that fast. But as you can see, more and more cells are coming to the tip of the needle with time. Yeah, there is another one, and probably soon another one will come. So there was definitely something going on at the very tip of the needle for magnetically presorted cells. So again, the cells which were before stuck in the magnetic column. What about the control cell fraction, so flow through? Here's the same needle, more or less the same concentration of cells in the cell suspension. And again, if you look at the tip, you cannot see anything. So basically, there is no attraction of cells at least due to magnetic interaction. So of course, all the cells are moving. This is just because we disturb the cell suspension with the needle. But as I said, no arrangement is taking place. Therefore, it only happens for magnetically presorted cells. Instead of describing it only with words, more or less, of course, we all like numbers. Therefore, we decided to quantify the total fluorescence at the needle tip and compare it during the first 5 minutes of the measurement. The total measurement was longer, but we only focused on the first 5 minutes, also because the cells start to bleach under the light with time. During the first 5 minutes, you can see that the signal from magnetically sorted cells, so the total fluorescence and the needle tip, constantly grew, while for all the other cell fractions, the control ones, it was basically fluctuating at the level of background. Therefore, we confirmed that we indeed see what we want to see. But the story is not yet complete, because I still need to quantify the force. In principle, it's realistic to be done, just because we know the magnetic moment of individual cell, and we can also calculate the magnetic field gradients at the tip of the needle. So it would be very nice to quantify the force from which magnetic field is acting on these cells, and to make sure that these numbers correlate with each other. So I come to my conclusions. So we show the possibility of creating ion biomineralizing encapsulins in model mammalian cells. We perform magnetic cell sorting without an external label. We show that the encapsulin particles represent nano-sized amorphous mekhemite. We independently confirm the ferromagnetic properties of cells by 2 individual techniques like vibrating sample magnetometry and ferromagnetic resonance. And also we showcase the manipulation of presorted cells with a magnetic needle, which brings me to the final outline that we successfully created physically plausible biomagnetic interfaces for cell manipulation. As I promised before, here is the one slide summary of my current project, where I don't right now at least work with genetically encoded particles. Instead, we come up with so-called semi-synthetic or semi-genetic approach, where we used fabricated nanoplatelets, synthetic antiferromagnetic particles with perpendicular magnetic anisotropy. We would like to change the surface of these synthetically synthesized particles and introduce some tags which would couple to ion channels I mentioned before, so the gates of ion influx. And if they are successfully coupled and you switch on the field, these platelets can disturb the ion channels and, in ideal world, open and close them. And we can see how much the ion influx changes. For this, we want to use the objects with a size about 120 nanometers created by nano-imprint fabrication. And what is interesting, of course, that in our project, we use pretty small platelets, which should be able to do the task we set for them. But in other studies, normally people use the structures which have much bigger size, around 2 micrometers. And on this slide, you nicely see the size comparison, how big the difference is. And of course, if we work with living cells, this difference is pretty much important. Of course, this work I described cannot be done by person alone. And I would like to sincerely thank my former supervisor, Professor Dr. Jill Westmeyer, and all other people, collaborators from Technical University of Munich, Herford Center in Berlin, National University Misses, Berkeley Lab, University of Oldenburg, and of course the team from the University of Duisburg-Essen, And first of all, Professor Dr. Ulf Widwald, who did a lot of magnetic measurement and also discussed with me constantly. I also would like to thank the funding organizations which made this study possible and also the funding for my current project. Unfortunately, I will not be able to join the Gathertown afterwards, but here is my email address. So in case you have comments or questions you won't be able to ask right now in Zoom, please contact me, and also for possible collaborations, I'm very open. And of course, I would like to thank you all for your attention and looking forward to your questions.
Thank you, Mariia, for the excellent talk. So if there are any questions, please raise your hand. So I have a question about the velocities of this. I mean, what velocities can be achieved, or is it important for cell manipulations to know the velocity?
Thank you very much for your question. So I would say that knowing the velocity would be a nice proof or a nice sanity check that our effect indeed can be explained by magnetic interaction. So if we quantify the force with which magnetic field is acting on each individual cells, and let's say we see that these cells actually supposed to move like several millimeters per second. Of course, I'm exaggerating, but nevertheless. But in fact, we, for example, see that they only move with the speed like a couple of micrometers per second. Therefore, we would need to think in this case what other force can be taken into account to make this equation of, force equilibrium brought it to zero. So this would be important for calculations, but actually for cell manipulation itself, I believe that maybe not the velocity would be important, but actually the threshold. So what would be the minimum field force, which is possible to actually start moving the cell, even if it's moving slowly. So imagine that you need to manipulate, let's say, not the cell itself, because moving the cells is maybe not so practical in vivo, although this we may speak about separately. But if we talk about subcellular compartments, like the ion channel I mentioned before, ideally one single particle should be able to somehow create a magnetomechanical distortion of the ion channel, which has some finite size as well. So the threshold of like how big the force can be created by this particle, this would be the crucial parameter we would need to know and, if possible, to maximize.
Thank you very much, Mariia. So, any questions? So, if not, thank you, Mariia, again for your great talk. And yeah, it's
thank you very much.
Yeah, that you're not able to join us in the Gathertown, and